Hello, everyone. This is chapter 12, which talks about cell cycle when one cell becomes two cells. Okay, the exact cell, they have to be exactly alike. Okay, so that's what I hope you guys can see this. So that's what this chapter is about. I have different type of cells. They become one cell becomes two cells. So. And then we'll talk about plant cells and animal cells and all other cells. Oh, he's starting the chapter with it. Okay, let's watch. Oh, he's showing you how the cells on the microscope, they're dividing and becoming two cells. That's right there. You see now one cell became two cells, and two cells can become four cells, and so on and so forth. Here they are. They're showing you this is, and then become a blastula, which we will not get into embryology portion now, later on, or when you take zoology, we'll talk about the embryology portion of it. And this is a blastocyst or blastula, and now you're seeing the gastrula. So we will not get into those parts. Just at the beginning, one cell becomes two cells. One cell become two cells. That's all we are going to get into now. Okay, I think it's okay. We can move on to the next slide. So maybe I should bring. I cannot read the heading because this is. Oh, sorry. Oh shoot. Okay. Now. Let me move you guys this down there. Sorry about that. Okay, so I hope I am. Okay, great. Most cell divisions, that's what it is, cell division, results genetically identical to daughter cells. Okay, that's what it is, exactly two identical cells. Most cell divisions results in daughter cells with identical genetic information. Yes, the DNA of both of them have to be same. It's not going to be different. The exception is in meiosis, which is totally a different chapter. You should not brought it up here at all. But anyhow, these are some of the terminologies I would like you to know. I put them up here for you guys. So cellular organization of genetic material, genome. Is if you have DNA, if you have a DNA like this, a strand of DNA, that segment is a gene. The other segment is another gene. The other segment is another gene. So each segment of the DNA is called a gene, which we'll discuss it later on in future chapters. So here you are, you're seeing chromosomes. Right here are chromosomes. And then the sister, a chromatin is the all of the nuclear material. I give you the definition here in the nucleus is called chromatin. Somatic cells, they are the cells which are inside of your body. Like my eye cell is a somatic cell, my skin cell is a somatic cell, my bone cells is a somatic cell. These are all called somatic cells, the cells of the body. Okay, a gamete are the sperm or egg. And in case of plants, it's called the pollen. For example, you all know pollen. You're allergic to pollen. Some of you are. Sister chromatid. Okay. So when the cell wants to divide, from one cell to two cells, the chromosomes have to divide. The way the chromosome, this one chromosome, I'm talking about this chromosome is dividing, then it becomes, this is one strand, if you would. And now here, look at it, is two strands. So this one strand right here is called a sister chromatid. And this one right here is called a sister chromatid. In this diagram, this is all a sister chromatid. In this diagram, this is called a sister chromatid. That's what they are, okay? And they are connected together at a blue dot right here, right here, right here, right here. They're connected together at centromeres. Okay, here are sister chromatids. Here is a chromosome. Here is a chromosome. They are connected in centromeres. And of course, that chromosome is made up of DNA, which I will talk about it, loops around the, here they are showing you the structure of DNA right here, and the centromere is chromosome, one arm, chromosome arm, and then the whole thing is called chromosome right here. But and then their sister chromatids, everything that I talked about it is right here. Eukaryotic cell division consists of mitosis and cytokinesis. Mitosis is pretty much a nuclear division. And then cytokinesis is the cytoplasm. Okay, and meiosis, again, this chapter should not talk about meiosis. We'll talk about it later. Do not worry about it now. What are phases of cell cycle? Okay, he's getting right into it. That's good. The cell cycle consists of mitosis, the M phase, mitosis, and cytokinesis. Interphase is not a stage of mitosis, and I hope you remember that. It's when the chromosomes are being duplicated. Interphase is about 90% of the cell cycle, can be divided into subphases. The interphase, these are stages of interphase G1, G2, S, and G2. S phase, that's where the chromosomes duplicate, okay? That's what they call it S because of synthesis. And then G1 is the first gap and G2 is the second gap, which I will talk about each one of them here in a minute. The cell grows during these three phases, but chromosomes duplicate during the S phases. Okay, right here, they showed you that these are the, if this is a clock, if you imagine this is a clock, 24 hours, 12 hours, whatever you like to think of it, there is only a small portion right here is where chromosomes and cytoplasm divide, okay? There is a big portion of the cycle is the G1. There is another portion of it, if we are dividing this, there's probably 1 or 2 hours devoted to mitosis, okay? And then out of 12 hours, let's say 12 hours, out of 12 hours, then divided by 12 divided by 3, about 4 hours, 3 to 4 hours is G1, 3 to 4 hours is G2, and 3 to 4 hours is G2S and G2. Okay, so anyhow, here are the stages of G2 interphase, a prophase, and a pro-metaphase. I'm not too crazy about this, pro-metaphase, and so on and so forth. But these are what is happening during the cell, and these are centrosomes, not centrioles. Do not confuse them with centrioles right here. These are centrioles. Remember, we talked about them. Centrosomes are the microtubules, which they are, you know, we talked about them at the chapter, God knows, 5, 6, somewhere in that neighborhood, that they can pull the chromosomes apart. So when the cell is dividing, is getting ready to divide, these centrosomes with the centrioles pairs, they divide, they go into opposite directions. And that is for animal cells, and they pull the chromosomes apart. So here they are, when the chromosomes are lined up in the middle of the cell, they call them metaphase. When the sister chromatids separate, okay, imagine my arms are each arm is a chromosome. So they are connected, that centromeres right here, they separate, okay, during what phase? Anaphase. During anaphase, they do separate. And then, of course, during telo, as you all know what telo means end, okay. During telophase, which the chromosomes, cytokinesis, and telophase, when the nucleus is going to start forming, and so on and so forth. And of course, it will follow by cytokinesis. I didn't talk about prophase. In prophase, I'm sorry about that. I didn't know your textbook is getting into it. During the prophase, the chromosome starts condensing and they start pairing up. You don't know it yet, but not in here. In meiosis, they start pairing up. But they all line up in the middle of the cell in metaphase. But during these, I will talk about aster centromeres and all of that a little bit later on. Anyhow, prophase, that's, I think it should come up. So G1, he's going backward. I don't know why, but no, I guess he's going through the cycle like this. I would explain it. G1, G2, G3, and then here is interphase, centrosomes with centrioles and centrosomes duplicated uncondensed. And here is a prophase when, you know, the centrosomes go on the opposites end. And here they're showing you, do not confuse these two terms, centrosomes and centromeres. So right here, kinetochore, I will show you, those are the protein molecules that the microtubules, non-kinetochore microtubules, they will attach to and then pull the chromosomes apart, metaphase, and right here, spindle. Very good. Okay, let's see what it's showing you. Oh, okay, that was quick. It showed you the three-dimensional chromosomes that they're being separated. Right here, anaphase, and then of course telophase. I'll talk about all of that. It's going to be a short one also. Okay. Nuclear envelope formation. Okay. And here are the pictures. They took it with, I don't know, not phase contrast, they stained them. Maybe they took it with phase contrast. So let's see what this one says.
This soccer player's knee will be as good as new in a few weeks, thanks to mitosis, a type of cell division that generates new cells for growth and repair. Let's move into a cell to witness the events of mitosis. Before a cell can divide, it must first duplicate the chromosomes stored in its nucleus. During chromosome duplication, several bubbles open up along the chromosome. Each bubble grows until it merges with an adjacent bubble. Each chromosome now consists of two identical copies called sister chromatids. Getting closer, we see that each sister chromatid consists of DNA wound around small proteins called histones. The sister chromatids begin to coil.
Histones, what can I write? Histones, okay? These protein molecules right here, right here, right here are histones. And of course, this is sorry. Come on. Okay, sorry about that.
Outside the nucleus, centrosomes that duplicated earlier move away from each other to opposite sides of the cell. Microtubules extend from the centrosomes, forming the mitotic spindle. Back in the nucleus, the DNA forms loops, becoming more compacted. These structures fold back on themselves, eventually condensing into a shorter and thicker chromosome consisting of two sister chromatids. As the chromosomes continue to condense, the nuclear envelope breaks up. The array of spindle microtubules is now extensive, and the chromosomes are fully condensed. Spindle fibers from each pole attach to protein structures located at the centromere of each sister chromatid. As the chromosomes are bound by spindle fibers from opposite poles, they move first one way and then another. The counteracting forces of the spindle eventually cause all the chromosomes to end up at the center of the cell, as if arranged on an imaginary plate. The sister chromatids are released from each other, each becoming a full-fledged chromosome. They are moved toward opposite poles of the cell, pulled along the spindle fibers attached to them. At the same time, overlapping spindle fibers that are not attached to chromosomes continue to lengthen, pushing the poles farther apart. Once the chromosomes arrive at their destination, they become less condensed. Two new nuclear envelopes form, completing mitosis, the division of one nucleus into two genetically identical daughter nuclei. The cytoplasm divides by the process of cytokinesis, forming two separate daughter cells. In your body, millions of cells divide every second, providing new cells for growth and for repair of damaged cells.
Right. Okay. Something I failed to mention. I'm sorry about that. At the beginning, I should have said if you cut your skin, mitosis is happening. You have a little baby at home growing taller and taller, mitosis is happening in his own cells. If you have a plant at home, your plant is growing taller, that's mitosis. So all of these are mitosis. As I said, you cut yourself or your skin grows back, not your muscles and not your nerve. But any other cells. And also, the cells of your body always are dying and growing back. For example, your skin cells, every 2 to 3 weeks, they die. And the new cells grow back. And that is mitosis. All the cells from inside of your mouth, the epithelial cells inside of your mouth, your anus, they're dying every 2 to 3 days. And new cells are growing back. So that is all mitosis. Or the rest of your body, again, except your muscles and your neurons, they are dying and growing back. Let's see what, and that's what mitosis is all about. In animal mitosis, they're showing you the chromosomes, and then of course they are dividing the divisions. Okay, that's anaphase. And now telophase, and then now you have cleavage failure. Yeah, they could have done it a little bit better. Anyway. Okay, the mitotic spindle, a closer look at this. So the mitotic spindle, the microtubules, which I already talked about it, that controls chromosomes movement during mitosis. Centrosomes, the microtubules organizing center, that's what the centrosomes are. And an aster, I've got to show you a picture. An aster is a structure that are that shorter microtubules that are also helping function of it. Some of them do not know, but some of them say that they're helping with pulling the chromosomes apart. The spindle includes the centrosomes, the spindle microtubules, and aster, okay? And kinetochore right here, that's what I was hoping to see that. So the aster right here, did I give you a definition of an aster? Aster, radial area of short microtubules, that's what I said, extended from each centrosomes. And the function of it, again, why they're there, I guess their support. That's the only thing I can think of it, their support for there. But anyhow, these are sister chromatids in animal assembly of the microtubules begins at the centrosomes. Yes, okay, so centrosomes are right here. and then the microtubules organizes, and as the kinetochore are proteins complexes associated with centromeres. So there are protein molecules called kinetochores right here, and they're showing you a transmission, TSM, transmission electron picture of it, and then TEM, I'm sorry, let's say TEM, transmission electron micrograph, not TSM, sorry, TEM. And then, so there are kinetochore protein molecules right here. And of course, the chromosomes are lined up in the middle. And these are the non-kinetochore microtubules and so on and so forth. So I'm showing a diagram. Again, he's showing you, okay, spindle formation during mitosis. Yes, so we saw that one too. What is cytokinesis? In telophase, genetically identical daughter nuclei have been separated. And then what happens in animal cells, you have cleavage failure in case of animal cells, and cell plates is in plant cells, okay? So what happens in animal cells, if the cell mitosis occurred, they start invaginating, folding inward, just like the diagram, beautiful. They took a, this is a SEM picture, scan electron micrograph of that picture is beautiful. And this right here is called cleavage farrow. Okay. In plant cell, the story is a little bit different. So the cell starts lying down little by little, cellulose, just like forming a dam on a river. They start lying down little by little, cellulose and protein molecules and all other goodies. They start adding up, and that is called cell plate. Here are the nucleus, the chromosomes, but right here, that's where cell plate is, and that's how cytokinesis animation. Let's see what it says.
In animal cells, cytokinesis begins with the formation of a cleavage furrow. At the site of the furrow, a ring of microfilaments contracts, much like the pulling of drawstrings. The cell is pinched in two, creating two identical daughter cells. In plant cells, cytokinesis begins when vesicles containing cell wall material collect in the middle of the cell. The vesicles fuse, forming a large sac called the cell plate. The cell plate grows outward until its membrane fuses with the plasma membrane, separating the two daughter cells. The cell plate's contents join the parental cell wall. The result is two daughter cells, each bounded by its own continuous plasma membrane and cell wall.
Very good. Okay, myosin and cytokinesis. Let's see what that is. So you saw cytokinesis. Here is a plant cell showing you cell plate right here. Okay, and of course, a prophase, metaphase, anaphase, and telophase. So whenever you see the cell plate, that has to be telophase. Whenever you see cleavage farrow, that has to be a telophase. So that's good information for your lab material, lab practical, and so on and so forth. Here they're showing you the cells. Okay, in bacteria, since they do not have nucleus, they divide differently. Okay, and the process in bacteria is called binary fission. Prokaryotes, bacteria, and archaea reproduce by type of cell division called binary fission, not fusion, fission. Okay, in binary fission, the chromosomes replicate, beginning the original replication, and the two daughter chromosomes actively more apart. The plasma membrane pinches inward during the cell in vitro. And right here, they're showing you the binary fission. So let's go back to the clock that we talked about. Here's mitosis right here. And then what happens during G1S and G2, in order for the cells, they don't go crazy and multiply and multiply and multiply, which you will see later on in the case of cancer, they will do multiply and multiply and multiply. So right here, G1 is a checkpoint, okay? So the control system checkpoints, there is one in G1, and there is one in here, and there is one in here at the beginning of the mitosis and at the end of the mitosis. So there are 3 checkpoints according to this. If you read different textbooks or maybe different scientific articles, they might say a different story, but you have to go by your textbook. So there are 3 checkpoints that they can stop the cell division. So cyclins and cyclin-dependent kinase, they are 2 protein molecules that they can, the cell cycle clocks. Let's read it what it says. 2 types of regulatory proteins are involved in cell cycle. One of them is cyclin, as I said, and cyclin-independent kinase, CDK. The activity of CDK, okay, the enzyme, rises and falls with change of concentration of the cyclin partner. Okay, the MPF, today I'm reading everything backward, MPF, maturation promoting factor is a cyclin CDK complex that triggers cells passages that pass the G2's checkpoints. Here they're talking about, I'm not too crazy about these things too much, but here is a picture of a cyclin, and here's a picture of the enzyme CDK. And when they are together, it is called MPF, okay? And then MPF breaks apart. When it breaks, it does not allow the MPF, does not allow the cell cycle to continue. And then what happens, it's in a G2 stage. And when that breaks apart, when the MPF breaks, shatters, then the cell cycle will continue on. And here is showing you the cyclin and MPF curves during the mitosis G1S, G2 mitosis again, and what is happening during those cells. And every cell in our body has a different clock. As I told you, the clock for our skin cells is 2 to 3 weeks, and the clock for our cells from our mouth to our anus is 2 to 3 days. So the cells in our body, they have different cells, different clocks they have. So here's a checkpoint, and then he's elaborating on these things, okay? So G1 and G2 checkpoints, and then the mitosis checkpoints. You can read it. What is an example of internal signal? An example of internal signal is that cells will not begin anaphase until all chromosomes are properly attached to the spindle at the metaphase place. This mechanism assures that the daughter cells have the correct number of chromosomes. What is an example of external factors? Okay, external factors that influence cell division include specific growth factors, very important. Later on, when I talk about animal cloning or stem cells and so on and so forth, you will see that term, the growth factors, that are scientists use growth factors to differentiate in a plate, okay, in a plate to differentiate one type of cell to another type of cells. Anyhow, growth factors are released by certain cells and stimulates other cells to divide. Here we go. I said that. Platelets derived from growth factors are P, D, G, F, and platelets are they're little protein fragments of cells, well, they're proteins as well. The fragments of the cells that allow blood clotting occurs in your body is made by blood cells fragments called platelets. In density-dependent inhibition, crowded cells will stop dividing. Okay, so what happens? A sample of human connective tissue is cut up into small pieces and in a petri dish, and enzymes digest the extracellular matrix, so the cells are by themselves suspension-free fibroblasts. Cells are transferred into culture vessels. PDGF is added. That's it. Remember that we talked about, PDGF, platelets. That's what that is. Platelets drive growth factor. Later on when you get into your life, anytime you read an abbreviation, please try to look up what that is, and that will help you to understand the material better. Anyhow, PGS is added to half the vessels, and here they are. You have a cells that you actually can, without PGF, with PGF, and with PGF, it differentiates to fibroblasts. These are fibroblast cells. Right here. Here they are, fibroblast cells. How is cancer being formed? The two concepts, anchorage and density-dependent inhibition. Cancer cells exhibit neither type of regulations of their divisions. Okay, so what is anchorage? If I have a plate just like right here, and I place a cell in here, and I put the right growth factors in here, drop them in here, they start growing, and they start growing, and when they touch a hard surface right here or on the bottom, they stop growing. Okay. And then density dependent inhibition. So when you have the same thing, they start growing, they start undergoing mitosis. So when they touch each other, they have receptors on the surface of the cell, these cells, okay? They have receptors on the surface of each other. They say, okay, stop. We are touching each other. That's enough. It's called density dependent inhibition. What happens with cancer cells? They do not have these anchorage and density dependent inhibition. They keep multiplying and multiplying and multiplying, okay? Here they are, they're showing you what happened, density dependent inhibition. As I said, they put a few cells, and then in case of cancer cells, and normal cells is converted to a cancer cells by a process called transformation. Cancer cells that are not eliminated by immune system form tumors. Okay, so our immune system is very important. We need to take care of it. Not stress, diet, so on and so forth. Masses of abnormal cells within otherwise normal cells. So we were born with cancer cells. Everybody was born with cancer cells. What happens, our immune system is supposed to take care of them and get rid of them. And when our immune system cannot get rid of them, then they grow excessive. If abnormal cells remain only at the original site, the lump is called a benign tumor, but it's a malignant tumor when they spread out through the body. It's called metastasis. Okay. So invade surrounding tissues and cells and so on and so forth. Localized tumors may be treated with high energy radiation, or which damage the DNA of the cancer cells. To treat metastatic, ooh, yeah, that's a tough word to say, in cancer, chemotherapies and that target the cells cycle may be used. Okay, here they are. The blood vessels will take the cancer cells to the rest of the body, and then begin. So here they are, the mitotic phases, and that's the end of the chapter. I hope